So, hello all. We will start the session today. So other people can join in between. So we will start the session. Is it my voice is audible? My slides are visible? Anyone? Is it my voice is audible?
Yes, sir.
My slides are visible. If you are not comfortable, you can put the message in chat box also. My slides are visible. Thanks, Ankita. So we will start the session. So myself, Ajit Kumar, a PhD research scholar from IIT Guwahati. In this session, we will see the week 3 live interaction session on the course Environmental Biotechnology. The course code is NOC 25 PT83. And I hope everyone has gone through the week 3 lecture of the Environmental Biotechnology. So this session will be completely recap of the week 3, and also we will see sample MCQs regarding the week 2 and week 3. So this is the agenda of this week 3 live interaction session. So before moving on to the course, I will introduce myself. Myself Ajit Kumar, joint degree research scholar, a research collaboration between IIT Guwahati and IIT BHU Varanasi. So I work in a lab of biochemical and environmental engineering, where our lab focuses on wastewater treatment in different industries. And especially my area of research is application of enzymes in wastewater treatment, especially textile wastewater. Moving on to my qualification, I've done BTech Biotechnology from Sastra University and MTech Industrial Biotechnology from NIT Surathkal, Karnataka. So my work experiences, I have worked in many pharma companies like Biocon Limited and Enzyme Bioscience, where I worked in R&D pilot plant and QMS, etc. So this is all about my personal introduction. So now we will move on to the week 3 recap, and I hope everyone has gone through the week 3 lectures. And in the week 2, we have gone through the what is species diversity, and we have seen the species richness and abundance, and we have seen biogeochemical cycle, and also the microbial oxidation reduction reactions, that is redox reactions. And we have started with microbial ecology, why we have to understand the microbial ecology and what are the ultimate goals of this microbial ecology. Then we have seen the isolation procedures, metagenomics and microarray basics concepts and where they have been applied. And also we have seen the MCQs of week 1 and week 2. So this session will be a recap of week 3 course and followed by the MCQ samples of week 2 and week 3. So we will move on to the session. So first, in the week 2 itself, we understood that microbial ecology and environmental biotechnology, they are tied to each other. That is, they are connected and they are merged each other. So how this environmental biotechnologist applies the concepts and tools of microbial ecology to manage environmental problems like cleaning the environment, controlling the pollution in order to produce bioenergies or biofuels, as well as producing valuable stocks or chemicals. So if we see the microbial ecology, So anyone can tell what is microbial ecology? The definition of microbial ecology? You can unmute your mic or you can post your answers in the chat box if you're not comfortable. So anyone can define the microbial ecology definition. Anyone? Microbial ecology. Okay. So microbial ecology is nothing but study of microorganism in a given community or in a given habit. So studying the microorganism and what is the ultimate goal of it? The first ultimate goal is to understand the microbial community. That is to know what kind of microorganism are present in a given community. Community means within a given area or a community has different species in it. So what kind of different microorganism that exist in a given ecosystem? So, which is self-organizing and self-sustaining assemblage of different microorganisms. So, if we take a microbial community, there will be a prokaryotes, eukaryotes. In eukaryotes, there will be fungi, algae, yeast, and different species. So, first we have to understand what kind of microorganism are present in a given ecosystem. So, after finding what kind of microorganism, then we have to understand what is its communication or interaction between them, between the species and also with the environment. So environment is nothing but environment is biotic or abiotic. Anyone? Environment comprises of biotic factor or abiotic factor, or is it both? So what do we mean by environment? Whether environment contain only biotic factors or abiotic factors or both? Yes, correct, Ankita. It comprises of both biotic and abiotic factors. And can you guys tell what is the any examples of biotic and abiotic? Any example? Abiotic means the environmental conditions. Yes, biotic means living organism. It can be microorganism, either fungi, plant, animals, any living organism. Abiotic represents environmental conditions like temperature, pH, soil, nutrient availability, etc. So, so how does this microbial community interact among themselves and also with the environment? So these are all the ultimate goals of microbial ecology. So as we have seen the basic definitions of environmental biotechnology, how we are going to use this environmental biotechnology to provide a good service to the modern human society. For that, we have to manage the microbial community. That is, these microorganisms are member of the community. So, how they are going to help us to provide service like preventing the pollution, controlling the pollution, biofuels, bioenergy, or to produce valuable stocks or chemicals. So, how do we manage them to produce these kind of application? We will see in the upcoming slides. So, some of the examples how we can manage this microbial community to support the human society. The first example is detoxification of contaminated water, wastewater, sludge, sediment, or soil. That is how do we detoxify or remove the toxic elements like dyes, etc., from the environment and convert into clean and harmless product. So for example, if we take the textile industry, we have dyes that is contaminated with water. So we cannot release these dyes into the environment without having a proper treatment. So what we do is we use appropriate microorganism in order to break down this toxic dyes and convert them into non-toxic form. Then only we can release them into the environment. So this is the example where microorganism is applied in order to detoxify the contaminated site. It can be either water, wastewater, sludge, soil, or sediment. So the next application is capturing of renewable resource, particularly energy and water. That is, renewable resource means it is a valuable resource that is converting some waste into valuable product like energy or water. For example, the organic waste can be converted into biogas like biomethanol or biofuels or bioelectricity also. This is like we are converting a garbage into gold. So we are capturing the gold like electricity or biogas from the waste. So the third application is sensing the contaminants or pathogens in the environment. Sensing means we can develop the sensor in order to sense these contaminants. Contaminants can be any emerging contaminants like heavy metals, antibiotics, or pesticides if it is contaminated in the environment. That is, we can use microorganism directly or their product that can be used as a sensor in order to detect any toxic heavy metals so that we can know the alertness of any danger in a given environment. And the fourth application is protecting the public from the dangerous exposure to the pathogen. That is, we have to suppose the wastewater not contains millions of microorganisms. That is, it can also contain harmful disease-causing microbes. So we have to protect it from the releasing into the environment, because if it is released into the environment, it can affect the whole aquatic ecosystem. So it is, for example, it is like a security check at the airport. So what happens? So the after complete checking only they will allow us to enter into the airport, right? So likewise, we will first analyze who are all the beneficial microorganism, who are all the harmful microorganism. Then only we will be we will be taking into the environment. So the harmful bacteria or virus will be caught, or it will not be exposed to the environment without appropriate treatment. So the last point is providing new molecules for diverse application. That is, new molecules like new medicine, new antibodies, or eco-friendly detergents or biodegradable plastics can be produced from the microbial products. So these are all the fields that we can use the environmental biotechnology field to serve for us, that is human society. So we will see examples regarding this in the upcoming slides. Before that, we have to understand why environmental biotechnology is so important and why it is feasible in the large scale production also. Because this environmental biotechnology, they can be taken into the large scale. That is, it can handle thousands of meter cube of wastewater per day, every single day. So it can be perfect for cities and industries since it can handle large volume of wastewater to treat. Then it can run continuously like 24 bar 7, treating the wastewater and breaking down this wastewater without much intervention. And the third major point is that they are largely self-controlling. That is, if we provide an appropriate conditions like nutrients or environmental conditions, if we provide them with the suitable condition, they will work like self-driving car for cleanup of pollution. And fourth point is they are economical, they are cheaper when compared to some other conventional treatment problems, so that they can be operated for a longer run and they are relatively simple to manage. If we are able to provide an appropriate nutrient media and environmental condition, So the managing these microbes is very easier than other conventional processes. So these are all the reasons why we need to be rely on environmental biotechnology. And moreover, they are eco-friendly and cost-effective. So these are all the important points that you need to take care, and it offers an advantage of going to the environmental biotechnology. So as we have seen, these microorganisms, if we manage them properly, we can use them for the human society, right? So one such example is superbug. So superbug is, as the word itself tells us, it is not a normal microorganism, somewhat special about the other. So why it is special? Because they are genetically modified. That is, their genes are modified through heterologous expressions. And one of the superbugs we all know, that is Pseudomonas petuda, that can degrade the hydrocarbons that are present in the petroleum waste. So you can think of like, for example, the main purpose is the main purpose is The main purpose is to treat the oil spills as a measure to control the oil pollution. So, if there is any accidental spills that occurs in the ocean, so we can use this superbug in order to treat this oil spills. So you can think of like it is a vacuum cleaner for oil pollution. That is, we will remove the oils from the water so that we can revive the aquatics ecosystem. So this is developed by Anand Chakrabarti in 1979. And we will see how this superbug, genetically modified organisms, are produced or constructed. So what happens here is, if you see the right hand side diagram, so we will take different bacterial strains and we will extract the specific plasmid. For example, the strain A, what kind of bacteria which has the capable of degrading the camphor related products. Why it is degrading the camphor related products? That is because of presence of cam plasmid, which has the capability of degrading this camphor group. So we will take the cam plasmid and strain B, which is a different bacteria that can degrade the octane related products because of the presence of oct plasmid. So if you see, so we will take this CAM plasmid from A and octoplasmid from B, and we will hybridize them, which is mix them and recombine the plasmid. So what we will get, the strain E, which is now have the capability of degrading both camphor and octane. Likewise, we will take the strain C, which has the capability to act xylol, and strain D, which has the capability to degrade the naphthalene-like products. Why we are taking these kind of strains? Because the camphor, octane, decane, xylane, and toluene and naphthalenes are the hydrocarbons that are usually present in the petroleum waste. So likewise, we will take the strain C, which has the XYL plasmid, and strain D, which has NaH plasmid, which has the capability to degrade naphthalene. Likewise, we will hybridize them and we will get the strain F, which have the capability to degrade both xylol and naphthalene type product. So what, after hybridization successfully, we will combine the strain E and F and we will get a superbug which has the broad capability of degrading vast hydrocarbons like camphor, octane, hexane, decane, xylane, toluene, and naphthalene based products, which are normally present in the petroleum waste. So, this is how the superbugs are formed through genetically modified microorganism through genetic engineering. And after knowing the basics of the genetic engineering of the superbugs, then we have to understand how it is helping in degrading this oil waste. First, what happens here is these microbes will attach to the substrate. Substrate is nothing but the oils. Then the microorganism will produce biosurfactant, which helps to dissolve and loosens the pollutant. It is like detergents to wash the dish plate, right? So what happens is this biosurfactants pulls all the oils, which enhances the contaminant solubilization and removal by increasing the surface activity. So once this biosurfactant has been produced, it helps in boosting the biopollutant bioavailability. That is, it is used to clump the oils, and after clumping the oil, this superbugs, as it had the appropriate plasmids, which we have seen, right? It has the capability to degrade all this camphor, octane, xylol, naphthalene based products, right? So once it is accumulated all the oil at once, it started to degrade this petroleum products, which includes octanes, naphthalene, xylene, toluene, and aromatic carbons. So this is how the cleanup of petroleum oil is done when it is accidentally spilled onto the oceans. If you have gone through my week one lecture, I would have explained the deep water horizon oil spill example where an accidental oil spill has been occurred in the deep water horizon. So where they have used this genetically modified organism in order to treat this petroleum oil spill. So this is one aspect of environmental biotechnology where microorganisms have been successfully applied. But still there are lot more challenges is there, but this is one of the example where this genetically modified organism has been successfully applied and it has helped the human society. And the next example that we are going to see how the environmental biotechnology is actually helping us is that the next concept is activated sludge treatment of wastewater. This is the conventional way of approach to treat the wastewater. It is often used for secondary wastewater treatment, where mainly they rely on removing the biodegradable organic matter. So, in order to understand this activated sludge treatment system, we have to understand what is the sludge. Sludge is nothing but settleable solids that are separated from the liquid during any textile process or any kind of industrial processing. So this sludge will contain a lot of microorganisms. So we can tell it as a consortium of microorganisms. So what it is an activated sludge. So it is an activated sludge process is nothing but an integral process which is used to treat the wastewater. How it is performed? So aeration will be provided in the form of air or oxygen that will be blown into directly into the raw sewage. So after providing the appropriate condition, it will help the microorganism to treat the wastewater aerobically. So anyone can tell the definition of aerobic process? So what do you mean by aerobic process? Aerobic process? You have heard aerobic and anaerobic, right? So what do you mean by aerobic process? Aerobic process means the word itself tells us yes, Ankita, it is right. So where oxygen is required, whereas in anaerobic, the process will be carried out in the absence of oxygen. So in terms of activated sludge, we will be giving the aeration, that is air or oxygen will be given to the system to treat the wastewater. So generally, it is one of these wastewater treatment system. It is that is secondary wastewater treatment system, and this conventional activated sludge will be having two parts. If you see the right hand side, we will be having one aeration tank or biological tank where the wastewater will be collected and sludge will be already present. And if we provide appropriate aeration condition, this microorganism will help in degrading or treating this wastewater. And once it has been treated, it will be taken to the clarifier where through sedimentation, we will be separating the solids and the liquid part. Where solid is nothing but the waste sludge. It can be treated further or it can be sent into the into the biological tank or aeration tank for recycling purpose. So then the liquid part will be taken into next wastewater treatment, which is tertiary treatment wastewater. If it is needed, if you are going for drinking water, you have to treat them through the sequential manner. So based on our requirement, we can proceed with the next stage of treatment. But as of secondary treatment is concerned, the activated sludge treatment is the most conventional and it is simple to operate to because you have to give aeration only so that the microorganism will be able to survive and treat this wastewater effectively. So this is the activated sludge where consortia of microorganisms they interact with each other and interact with the environment. If the environment here is appropriate temperature, giving aeration, so these are all determines the microbial consortia performance, which helps in treating this wastewater. So this is the second example where the environmental biotechnology has been successfully applied in order to treat larger volume of wastewater. And the third example is anaerobic digestion of sludge, where first we have to understand, as the name suggests, it is an anaerobic digestion. That is, the process will be carried out in the absence of oxygen and digestion of sludge. That means, so it is going to be a sludge. Sludge is nothing but the consortium of microorganism. So it is a biological process in which the organic solids are decomposed into stable substance. So if we define this anaerobic digestion process, it is a series of biochemical reaction where the bacteria which helps in degrading this organic matter. It can be any source, like it can be a protein, it can be a sugar, any kind of organic matter into a gaseous mixture. For example, methane, carbon dioxide, hydrogen, or hydrogen sulfate, which is in the presence, in the absence of the free oxygen. So if you see the right hand side, so it will be taking place in a sealed tank, which prevent the oxygen entering into the tank, and it will be capturing the gas that are produced during the reactions, and the digested biosolids will be collected at the bottom. So these gas can be used as a fuel for future purpose. So this is how under anaerobic condition this organic substrate is converted into a valuable product. So we will see how it is done. So this anaerobic digestion process occurs through multiple steps. It involves not a single microorganism, different types of microorganisms. So for example, these are some of the examples that participate actively in the anaerobic digestion. Anaerobic digestion. That is Proteobacteria, Chlorofluxi, Bacteroides, Clostridium, Methanobacteria, and Thermoplacenta. So these are the some of the microorganisms that are actively participate in the anaerobic digestion. And if we see the process, so what the reaction behind it, it comprises of four stages. So first is hydrolysis, second is acidogenesis, third is acetogenesis, and fourth is methanogenesis. So these are the four basic steps, and here we could see that in hydrolysis process, fermentative bacteria will be participating at different levels, converting the complex biomolecules into monomers. Then, with the help of the acidogenic bacteria, it converts into volatile fatty acid. Then, through acetogenic bacteria, it will be converting into acetates, hydrogen, and carbon dioxide. And eventually, with the help of the methanogenic bacteria, it converts into methane and carbon dioxide, where methane is used as a biofuel. So, if we take the simple example of glucose, through this anaerobic digestion process, it will be eventually converting into carbon dioxide and methane. So here we could see that methane and carbon dioxide is produced. So this organic material such as glucose is biochemically digested and it is converted eventually through process of four phase, which is hydrolysis, acetogenesis and methanogenesis. So these are all the four important steps that has been carried out in the anaerobic digestion. And we will see these steps one by one. That hydrolysis means it is the first stage in the digestion process where it breaks down the complex polymeric structures. It can be cellulose, starch, proteins, etc. So what it does, it breaks down the complex structure into simpler structure, that is monomers. For example, if we take the protein, it will be digest them into amino acid. If we take starch, it will be converting into corresponding monomers of carbohydrates. So such as glucose, fatty acids, and amino acids. So what uh, hydrolytic bacteria has been used for this hydrolysis, and these bacteria are capable of producing hydrolytic enzymes, that is amylase, cellulase, lipase, protease, and pectinase. So these are all some of the enzymes that is produced by the microorganism in order to break down the complex polymeric structure into simpler units. So this is the first step of the digestion process where the complex polymeric structure is converted into simpler units. So the second stage is acidogenesis. As the name itself suggesting as that acidogenic bacteria will be involved in it, and moreover acids, that is volatile fatty acids, will be generated at the end of the reaction. So we will see what happens here. So the product from the first stage, that is hydrolysis stage, will be further broken down into acidogenesis by using acidogenic bacteria. And what happens? They will be partially transformed into short chain volatile fatty acids. That is acetic acid, propionic acid, formic acids, and lactic acid. Sometimes it also changes to alcohols and ketones. And apart from this, we can observe a small traces of carbon dioxide, hydrogen, and alcohols are also produced as a byproduct. So if we see the second stage where we could see that these monomers are converting into short chain volatile acid, as well as sometimes it converts into some acetates and H2 and CO2 as a very less amount. So the third stage is acetogenesis, where acetogenic bacteria will be involving in it, and here the leftover volatile acids will be completely hydrolyzed to acetates, carbon dioxide, and hydrogen. So this is the third stage where acetogenic bacteria play an important role in converting long chain fatty acid into acetate, carbon dioxide, and hydrogen. And at the last stage, we can see that these acetates will be converting into methane and carbon dioxide. So this is the last stage where methane is produced from all the intermediates. So it can be leftover volatile fatty acid or leftover acetate. So these are all being converted into methane using methanogenic bacteria. Okay, so these acetates and H2 are converted into CO2 and methane by two type of bacteria. One is hydrogenophilic bacteria, which helps in converting the hydrogen into corresponding methane and CO2, and acetophilic bacteria. So these kind of group of bacteria will help in converting this acetate into carbon dioxide and methane. And acetophilic bacteria also helps in converting the CH4 and CO2, while hydrogenphilic bacteria converts H2 and CO2 into methane. So this is the process, and at each stage, we could observe that different types of bacteria will involve in converting them into methane and carbon dioxide. So if you see the last stage, it will be performed in the strict anaerobic condition, where the acetate will be converting into methane. So this is the third example where the environmental biotechnology has been successfully applied in converting the organic waste into a valuable product like biomethane. So till this, do you have any questions or any doubts? So till now we have seen how environmental biotechnology provides service to the modern human society and why environmental biotechnology is so important at the industrial level. And we have seen three examples where environmental biotechnology, that is microorganism, has been successfully applied. First is superbug, which helps in treating the oil pollution. Then we have seen activated sludge treatment system. Third, we have seen anaerobic digestion of sludge, where organic waste is converting into a valuable product. So these are all the three examples we have seen so far. So far, any doubt? Okay, Ankita. So then we will move on to the next topic, that is the areas attention required in the environmental biotechnology. So these are all the areas that has been focused so far in the environmental biotechnology. For example, in the water system, as we know that poor quality source need substantial treatment to eliminate the public health risk, unpleasant taste, odor, and discoloration. So normally, if we consider the wastewater, these are all the factors that we have to consider. That is, unpleasant taste will be there, orderness will be there, discoloration, high amount of dyes or high amount of BOD, COD will be present. So that these are all the some of the factors that are concerned before releasing into the environment. So the wastewater needs to be treated properly before discharging them into the environment or for usage of clean drinking water. So in water area, the environmental biotechnology has its foot, and also in energy, as we have know that the fossil fuels are destroying and we have to use the resource efficiently. So if you see, the future source must shift from fossil fuels to renewable ones, and we have seen that environmental biotechnology can help in converting this organic materials into bioenergies. So in energy field also, this environmental biotechnology can be used. And in environmental quality, as we have seen increasing population, urbanization, and industrialization, these are all high-end the need to apply the environmental biotechnology to improve the environment quality, along with capturing the valuable resources or valuable chemicals, etc. So in environmental quality also, it plays an important role, and especially in the human health, infectious or pathogen causing bacteria or any microorganism is the main cause of death worldwide, right? So in order to uh, silence the pathogenic microorganism, we will be using a beneficial microorganism in order to tackle the human health. And also security, terrorist threats to disseminate pathogens, that is bio weapons, they have been using as a bio weapons poses a great danger. So these are all the areas that needs attention in the environmental biotechnology. So in order to understand this environmental biotechnology, the microbial ecology is one of the important topic. And as we have seen, the microbial community plays an important role. And as we have seen, the ultimate goals of microbial ecology, that is to know what kind of microbial organism are present, and second, how do they interact among themselves with the environment. So microbial community is nothing but it is a self-assembly of multiple organisms. It can be either prokaryotic or eukaryotic organism in an environment to assist the ecosystem. So in order to understand the self-assembly of microbial community concept, we have to understand these four questions. And if you are able to answer these four questions, then we will be clear in the concept of microbial community. The first question is we have to understand what is a principle, underlying principle of this self assembly. So, what is the principle behind it? We have to understand it first. Then, what community structure is optimal for biotechnological application? That is, we have to find a best team among the others to help for the biotechnological application. In this, we have to find a correct mixture or a proportion or consortia that can work for a particular case. For example, if you want to treat textile wastewater, you have to find a good team or good microbial community that has the capability of degrading the toxic dyes. So the second point tells us to choose a better or best team among different community. And third one is what environmental condition triggers the assembly of the desired community with its desired function. This means after you find a better team or a best team among various community, you have to give a correct condition. Condition means like environmental condition like pH, temperature, appropriate condition needs to be given, and also you have to give appropriate nutrients so that it can attain the desired function and community. So you have to give appropriate environmental conditions so that it triggers the desired function. Then we have to understand all these are conditions and outcomes that we have given before is predictable and reproducible and controllable. So we will be training them in order to produce the particular molecules to tackle the environmental problems. So in order to understand the self-assembly of the microbial community, we have to ask these four questions. So first is you have to understand the self-assembly of the microbial community by diving into the principles why it has self-assembled in such environment. After understanding the principle, next you have to find a best team among them, and then you have to provide appropriate condition to that best team, and whether it can be predictable or it can be reproducible or it can be controllable, you have to see. So if you are able to manage these, we can manage this microbial community. Then, as we have already seen, managing the microbial community is one of the important concept where we could see it is nothing but it is a creating a technology so that the microorganism will work for us and we will also work for them. How they work for us to produce a valuable products like bioenergy or biochemicals, and how do we work for them? It is by giving the appropriate conditions like temperature, pH, and also giving correct nutrition source. So by giving appropriate environmental condition, so it creates a win-win situation for both microorganism and the human, so that we can manage the microbial community very well. So this is how you have to understand and manage the microbial community so that it will be beneficial for all the species. So, in order to manage this microbial community, you have to understand these five principles, and we will see this each principles in detail in the upcoming slides. And the first principle is that you have to aim for big benefit, and second is you have to develop and apply more powerful tools to understand the microbial community. And third is follow the electrons. And fourth is retain the slow growing biomass. And fifth is integrate all four in order to manage the microbial community well. So we will see one by one each. At least we will see the first two in this session, and rest of we will see in the next week. So first we are moving on to the aim for big benefit, which is nothing but Environmental biotechnology aims always for a big benefit, that is, especially in the water and energy field, so that the wastewater can be either it has what are the component, it will have water, so we can treat this water and we can use it for many purposes like we can use it for agricultural purpose, for gardening, or for their own industrial reusage, or it can be used for drinking purpose. And the sludge that is produced can be converted into valuable products like bioenergies, or their biosolids can also be converted into fertilizers. So this is how the wastewater is turning into resources. This is how we have to extract the benefits out of it. The biggest benefit will come when environmental biotechnology is directed towards the challenge of the society. And if we see what are the challenges, the main challenges is water and energy sustainability. For example, waste to resource, where the wastewater, sludge residues, and other kind of waste, now it should be viewed as a resource, so that we can get help from the microorganism, and that will work for us to produce valuable products like energies, in some cases biochemicals, or valuable chemicals also can be produced. This is how you should aim for big benefits, and we will see one example where this waste has been converted into resource. So if we take one such example is that biodiesel, where this is one of the article where they have tried to optimize the biodiesel production from the waste, that is, they have used waste in order to convert into valuable resource, which is biodiesel. So what they have done is they have taken the municipal sewage waste sludge after settling down, separating the liquids from the solids. They have undergone filtration followed by centrifugation and drying, and at last they try to extract the lipids as much as possible. And we all know that in order to produce biodiesel, we need to lipids in order to convert them into biodiesel. So the lipids, from lipids, we will extract them through oxalate extraction using appropriate solvents. So once the lipids has been extracted, we will do trans esterification process, where we will be providing some reactants like catalyst methanol along with these lipids. Once it has reacted with methanol and catalyst and lipids, it will convert into biodiesel. So here we could see that by adding methanol, catalyst, and from the lipids extracted from the oxalate extraction, it is converting into biodiesel. So this is how a waste like municipal sewage sludge is converting at last to biodiesel. And we all know that biodiesel is a renewable source, and it has finds its application now, and we are all moving into eco-friendly manner. So this is how we have to aim for big benefit, that is waste should be converted into bioresource. So this is the first principle, that is aim for big benefit. And the second one is develop and apply more powerful tools to understand the microbial community. That is, you have to build a more powerful and modern tools so that we can understand the microbial community very well. And why do we need such powerful tools? Because the microbial ecosystem are incredibly complex, and they have different species. They interact among themselves. Am I audible? Okay. Some connectivity issue was there. So we will continue the lecture. So we have been seeing why we need to go for a powerful tools. As I said, these microbial ecosystems are incredibly complex and they have different species. They interact among themselves, also with the environment. They share their nutrients and adapt to various environment. So such complex understanding or among themselves, we still don't understand much on the microbes at molecular level. So we have to depend on the more powerful tools in order to understand their community. So with the existing tools, we are still in the lagging phase and more research needs to be conducted for developing more powerful tools. And in order to develop a new molecular tools, you need to have three important criteria for devising any new molecular tools. That is, first, high throughput and depth of the information. This tells us that we have to upgrade the things as we have been upgraded from single camera to 3D, right? So here also in tools we have to upgrade and which needs to give the depth of the information. And next one is quantitative. It should give numbers, quantitative data in a very minimal time. And third, the tools must reveal the structures and functions parallelly. So we have to devise the molecular tools in such a way that in addition to the structural information, it also should reveal the functional information so that we can understand who they are and what they are and why they are communicating among themselves. So we could answer a lot of questions if we are able to succeed in attaining these three criteria, which is high throughput, quantitative data should be attained within minimal number of time, and it should also reveal structure and function in parallel. And if you see the right side diagram, nowadays gene editing has been comes into play where CRISPR has been used extensively, and also plasmid assembly like golden gate assembly, high throughput automation has been play an important role, and bioinformatics, which is a powerful tool in order to understand this microbial community, has been used to understand this functions. And as we have seen, these has been applied in different applications like from pollutant degradation, bioremediation, biosensor, and compound production. So these are all the tools as of now they have discovered, but in order to develop a more powerful tools, we have to keep in mind these three aspects before devising any molecular tools. And as we have seen how the bioinformatics tools is more powerful, which helps to understand the microbial community beyond genomics. So as we all know, transcriptomics, that is as the name suggests, transcriptome. Transcription means anyone know the definition of transcription? Transcription, we have studied in molecular biology. Yes, Ankita, it's correct. Transcription refers to converting the DNA into RNA. So the product is RNA, right? So transcriptome means it is the study of mRNA as an indicator of gene expression. Whereas proteomics means it is a study of proteins or their enzymes so that we can characterize and quantify them as it relates to the cellular level. So proteomics is nothing but study of proteins and enzymes. Metabolomics means studying the metabolic intermediates of cellular function. For example, if you want to study how glucose is converted into acetate, what are all the intermediate steps and what are all the enzymes that catalyzes these intermediate steps? These are all comes under metabolomics. So these three fields play an important role which helps in understanding the microbial community beyond genomics. So, as the term suggests, transcriptomics means studying the mRNA as an indicator, whereas proteomics is study of proteins or their enzymes, whereas metabolomics means studying their metabolic intermediates. So, these are all the three definitions and powerful definitions that you need to remember. That you need to remember. So why do we need to study these fields? So if we able to combine this data from the transcriptomics, proteomics and metabolomics, a researcher can gain the understanding of how genes, proteins and their metabolites can interact to drive the any cellular process. For example, a researcher wants to study how changes in the genetic level, that is gene expression, how a particular gene, for example, cellulase gene is responsible for catalyzing the hydrolysis process of cellulose, right? So how does this gene expression leads to changes in the protein level, in the proteomics, that is proteomics, and ultimately how it affects the metabolite concentration, metabolomics. So this is how we integrate these bioinformatics tools to understand the overview of the cellular process. So one such powerful tool that we are going to see is SIP, which is nothing but stable isotope probing, which is nothing but uh, it is like a GPS tracker. We all know GPS tracker, like which tracks the used for tracking the any vehicle or anything. So likewise, to track or to trace the uptake of nutrients in the biogeochemical cycling by microorganism, we will be using this stable isotope probing, which is one of the powerful tools that have been used in the microbial ecology, where what happens is that I will explain the simple way. First, for any microorganism, you need substrate or food to survive, right? So what happens here is we will be labeling the substrate with the isotope, which is either C13, N15 or O18. So we will label the substrate and we will inject into the microorganism so that the microorganism heats and these isotopes will be entering into their DNA so that we will track the path where these substrates are traveling. So this is how this stable isotope probing is helping in analyzing the uptake of nutrients in biogeochemical cycle. So if you see the right hand side, for example, any environmental sample, we will take one soil sample and we will first label it with either C13 or N15 or O18. This is the sampled labeled sample and one we will take as a control where unlabeled will be there. So first after adding the isotope, so it will be binding to their DNA. So we will extract their nucleic acid. Then through centrifugation, that is cesium chloride gradient. This is one kind of centrifugation that will be done and we will be analyze the nutrients. How this nutrients are uptaken by the microorganism in the given ecosystem. So this is how the SIP works and it helps in tracking the uptake of nutrients so that we can build the biogeochemical cycle. So what happens here? The substrate will be enriched with heavier stable isotope, that is either C13, N15 or O18, that will be consumed by the microorganism that needs to be studied. So once the biomarker with the heavier isotopes are incorporated into the microorganism, then it will be separated from the biomarker containing more naturally abundant lighter isotope by isopenic centrifugation. So this is how this stable isotope probe helps in tracking the nutrient in the biogeo cycle. So this is one of the powerful tool that has been helpful in understanding the microbial community. And the other important tool that we have come across is nanosims, which is nothing but nanoscale secondary ion mass spectrometry. So it is nothing but a super microscope for viewing the atoms. So it is a more powerful tool that is used to map the distribution of elements and isotopes at the nanoscale. So if for example, in the right hand side, we could see in order to understand the soil root system, we will be taking this soil matrix and will be bombarding the ions into them. So that what happens? The secondary ions will be released and this will be deducted. And what data it gives? It gives numerous data like what is the distribution of chemicals and how it identifies what kind of transformation it is undergoing at the molecular level. We can identify them. So it is primarily used to map the distribution of elements, and also it allows the researcher to visualize and quantify the elemental composition. So how much composition like carbon, hydrogen, nitrogen. So this elemental composition can also be done with a very small amount of volume. That is very small quantity of sample is enough in order to examine the interaction with the environment. So how does it helps? It helps to show where the nutrients like carbon, nitrogens are stored in the soil root system, and also how it reveals how a single bacteria can interact with these neighbors as well as with this environment. So it answers the question what and where. So where it is interacting with the soil root system and what kind of bacteria it is helping for this microbial ecology system. So it helps to understand the interaction between them and study their single cells also. So these are all the two important powerful techniques that has been developed as far as the second principle concerned, that is develop and apply more powerful tools to understand the microbial community. So as we have talked about the powerful tools, so as we have know that the microbial ecology and environmental biotechnology are tied to each other, right? So to create and to create and understand the microbial community interaction, we have to understand these three themes. If we are able to understand these three themes, we could analyze and understand the microbial ecology very well. So the first is you have to understand the powerful analytical tools that are prevalent in the microbial ecology. For example, as we have seen the power of like SIP, stable isotope probing, and also nanosims. So these are some of the tools that helps us to understand the microbial ecosystem. Then we have to understand the integrated omics approaches. Omics means proteomics, metagenomics, and transcriptomics. So with the help of these integrated omics approach, not only with the both powerful analytical tools and omics, the basics or the theory behind it or what process it drives the research. If we could understand these three aspects so that one can understand the microbial ecology and one can devise the concept of environmental biotechnology. So these three peak that we have to remember to develop and understand the microbial ecology. And as the last part, we have to understand and know what are all the criteria for a new molecular tools. As we have seen, the molecular tools should be high throughput. That is, it should generate large amount of data in a minimal number of time. For example, in the traditional or in long pack, the DNA sequencing will be done manually and it takes several days to sequence a very minimal base pair. Whereas now, with the advent of next gen sequencing, this DNA sequencing has been done within a minutes to hours. We can sequence millions of DNA base pairs within a minute, right? So this is how we have to develop the tools which should have high throughput. And next, the output that we are getting should be highly accurate quantitative results. So that enough to provide the informations that sorts out the lead actors from the bit players and to find the important organisms and reactions even when others dominate in numbers. That is, it should work accurately. Even though, if you see, in a community, there will be millions of microorganisms. Even though it has millions of microorganisms, it should accurately detect the most important microorganism that we are targeted on. And it should give an appropriate, correct quantitative data. And third important aspect is it should provide the type of information that should reveal both structure and function parallelly so that they are connected to each other. So this is how you have to remember the criteria for new molecular tools. The first one is it should have high throughput. It should give quantitative data. Third, it should reveal the structure and function parallelly. So to devise any molecular tools, you have to keep in mind these three important points before devising any powerful tools. So these are all the topics of the week 3 lecture of the environmental biotechnology course. So as the next session we will be seeing, now we will be seeing MCQs, sample MCQs. So before going on to that, if you have any doubts, you can unmute the mic or you can post your queries in the chat box if you have any doubts. So these are all the topics that has been covered in the week 3 of the environmental biotechnology course. Among 5 principles, we will see 3, 4, 5 in the next lecture. So as of now, is there any doubt? Okay, okay, Tuil. So if there is no doubt, can we move to MCQs, sample MCQs? So this should be an interactive session. So I will first give the question. You just take time and answer those in the chat box, or you can unmute the mic and you can answer the MCQ. So we will move on to the first MCQ. which is with respect to the pond's ecosystem, identify the correct option about the primary producer. Option A, aquatic plants. Option B, insect or fishes, etc. Option C, anaerobic bacteria. And option D, methanogenic archaea. So I will repeat the question. With respect to the pond ecosystem, identify the correct option about the primary producer. You have to find the primary producer in the pond ecosystem. So what will be the correct answer? Option A, option A, option A, it is an aquatic plants and algae. Okay. Any other answer? So we will see the answer. Yes, it is correct. Answer option A, aquatic plants and algae. So, so what do you mean by primary producers? Anyone? Primary producers are, what do they do? These primary producers? Yes, they are autotrophs. What they do? Yes, what they do? They will produce their own food through photosynthesis using sunlight to convert carbon dioxide and water into organic compounds. So, if we take the pond or aquatic ecosystem, the role is primarily filled by aquatic plants and algae, which contains chlorophyll and they can perform photosynthesis. So, insects and fish belongs to which category then? Insects, fish belongs to which category? Whether it is a primary producers or consumers. In consumers, is a primary consumer, secondary consumer. So what role they play? Insects, fishes, etc. They are they are consumers. They are primary or secondary consumers. They are not producers. They obtain the energy from the microorganism. So this anaerobic bacteria, these are also, they do not produce their own food through photosynthesis. So they are not primary consumers. They are not primary producers. They are also consumers, as well as the methanogenic bacteria, which is also an anaerobic bacteria. So here also there is no photosynthesis involved. So the this led to the option A, aquatic plants and algae, which is the primary producers. So we will move on to the next question. Which one of the following statement are true with respect to the Gaia hypothesis? I will repeat which of the following is true with respect to the Gaia hypothesis. The Earth is single sentient superorganism. Option B, the Earth seeks to apply selective pressure to enhance the growth of specific organism. Option C, the Earth's abiotic system behaves as a single independent organism. Option D, the Earth is attempting to return to the homeostatic state by eliminating all organisms. And option E, the Earth's biotic and abiotic system interact to preserve life and maintain self-regulation. So among these statements, which is true about the Gaia hypothesis? So what is the answer? Which is true regarding the Gaia hypothesis? Option A, okay. Any other answer? Varsha, option A, okay. Any other answer? So among this statement, which is true regarding the Gia hypothesis? This has been discussed in the week 2 lecture, I guess. The Gia hypothesis. Option E. The Earth's biotic and abiotic system interact to preserve life and maintain self-regulation. Okay. Okay. Now we will see the answer. The correct option is option E. The Earth's biotic and abiotic system interact to preserve life and maintain self-regulation. And why it is so, we will see. As the Gaia hypothesis proposes that the Earth and its living organism, they are interconnected system. Interconnected system tells us that both the abiotic and biotic play an important role in regulating the condition to maintain the life on the Earth. So it also suggests a dynamic balance between the different life forms. So this is what the Gaia hypothesis tells. So the correct answer is option E. So we will discuss the other options like so Earth is a single sentient, sentient superorganism. Earth is not a single; it is a complex self-regulating system, right? So option A won't be there. Option B is Earth seeks to apply selective pressure to enhance the growth of specific organism. No, always Earth maintains the overall condition for favoring the growth of life, right? So it not actively selects the specific organism for enhanced growth. So it tries to maintain the balance between biotic and abiotic factors. And option C, the Earth's abiotic system behaves as a single independent organism. No, it is not only abiotic system, it is both biotic and abiotic. The interaction between the biotic and abiotic system is addressed in the Gaia hypothesis. And fourth is Earth is attempting to return to the homeostatic state by eliminating all organisms. It is not returning to homeostatic state by eliminating organism. The Earth is stable and it is an habitual, right? So it doesn't involve in elimination. Instead, they try to balance the dynamic balance between the different life forms. So the correct answer to the Gaia hypothesis is the Earth's biotic and abiotic system interact to preserve life and maintain self-regulation. So this is the answer for the second question. We will move on to the third one. The third question is the nutrients in soil are made accessible to the plant by the activity of producers, primary consumers, secondary consumers, gentrivores, and autotrophs. I will repeat the question. The nutrients in soil are made accessible to the plants by the activity of which either it producer, primary consumer, secondary consumer, gentrivores, or autotrophs. Option D, which is gentrivores. Okay, we will see. Yes, it is option D, gentrivores. Can anyone tell what is gentrivores? What kind of organism it is, gentrivores? Dentrivores are that converts complex decaying organism into soluble form or uptake by plant. Exactly correct, Ankita. Yes, dentivores are organisms that break down the dead organic matters like decomposers, bacteria, fungi, which releases the nutrients back into the soil, making them accessible to plants. Correct, Ankita? So we will see what are all the other options. Producers, we all know that producers are they create their own food through photosynthesis. So they use it for themselves. And primary and secondary consumers are nothing but it is an herbivores, which is primary consumers are herbivores, which is that eats plants. So they do not directly involve in making the nutrients that are available in soil. Whereas the secondary consumers, these are all carnivores, so they eat the primary consumers. That means they are also not directly involving the nutrients that are accessible to the plants. And autotrophs, they are actually nothing but it is one of the type of producers. So it is also not directly accessible to the plants. The nutrients are not accessible to the plants. So the correct option is the dentivores. So we will move on to the next question. Prokaryotic microorganisms are the most genetically diverse organism on Earth. Which of these is not a probable explanation for this trend? I will repeat the question. Prokaryotic microorganisms are the most genetically diverse organism on Earth. Which of these is not a probable explanation for this trend? Option A, they reproduce quicker and thus evolve quicker. Option B, they are small and easily evade most predators, persisting longer in the environment. Option C, they were the first life forms to evolve, hence they had been evolving for longer time. And option D, their DNA replication machinery is in general less robust and tends to create more mutation. Option E, they can perform horizontal gene transfer relatively easily. So I will repeat the question. Prokaryotic microorganisms are the most genetically diverse organism on Earth. Which of these is not a probable explanation for this trend? Your answers? Option D, option B, option D, option B. Okay. The correct answer is option B. They are small and can easily evade most predators, persisting longer in the environment. So this is not the probable explanation for prokaryotic microorganisms are most genetically diverse organism on earth because there are prokaryotic and eukaryotic organisms that prey upon other organisms. Even higher eukaryotes can feed on aggregates of prokaryotic organisms. So in the option, this is not only applicable for prokaryotes, it is also applicable for eukaryotes. That's why this is wrong explanation. That's why the answer is B. So we will move on to the next question. Majority of biotechnology processes relevant to environmental application make use of microorganism because microorganisms have very specific growth requirement. Microorganism can easily adapt to variable condition or microbial biocatalyst is very expensive. Option D, microorganisms are quite small in size. Option E, microorganisms grow slowly. So which of these reason applicable to environmental application that make use of microorganisms? I will repeat the question. Majority of the biotechnology process relevant to environmental application make use of organism because option B. What is the option B? Microorganism can easily adapt to variable condition. So we will see the answer. Yes, it is correct. Option B, microorganism can easily adapt to variable condition. So we will see the explanation. So microorganisms are incredibly diverse and it can thrive or it can survive in wide range of environmental conditions such as extreme temperature, pH level and nutrient availability. So this adaptability make them ideal for biotechnological application, especially environmental biotechnology, such as to clean or control pollution. So, okay. So have you heard of this extremophiles? So Extremophiles are nothing but they can survive in extreme conditions. So, what do you mean by thermophilic bacteria then? Anyone? Thermophilic bacteria. So, these are all extremophiles, right? So, what do you mean by thermophilic bacteria? Thermophilic bacteria are nothing but they can, yes, it can grow in higher temperature. Likewise, there are different groups of bacteria are there, which is acidophilic, alkaliphilic, which can grow in acidic condition or alkaline condition. Though, so if a microorganism can able to adapt to different environment, then it can be widely used for environmental purpose because in environment there is a drastic changes in the environmental condition. And we will see the other options too. So microorganism have very, if they are having a very specific growth requirement, we cannot use it for wide range of application. So adaptability to variable condition is an important factor while considering for environmental application. So we know that microbial biocatalyst is normally cost effective, less expensive when compared to other conventional processes, and microorganisms are quite smaller in size. Yes, it is, the information is correct, but the application to the environmental field is not correct because if you see, we are not seeing the size. If you are considering the environmental application, instead of we are seeing the higher surface to volume ratio. If it is have higher surface to volume ratio, then we can select this microorganism. And the microorganism grows slowly. If they are growing slowly, then their growth rate is very slow. Then their lag phase is very high. So we cannot directly apply them in the environmental condition directly. So these are all the wrong information. These are all incorrect statements, and the correct answer is microorganism can easily adapt to variable condition. So the next problem is, why is the conventional definition of species not adequate for microorganism? Why is the conventional definition of species not adequate for microorganism? Option A, microorganisms have much greater genetic diversity. Microorganisms reproduce asexually. Microbial ribosomal structures is different. Microorganisms are too small for their physiology to be studied. Microorganisms evolve too quickly. So, which is the correct answer among this? Why is the conventional definition of species not adequate for microorganism? Option B, which is microorganism reproduce asexually. Option B. Okay. We will see the answer. Yes, it is correct. Option B, because the conventional definition of species is the group of organism in which two individuals of the appropriate sex or mating types can breed to produce the fertile offspring. So the correct answer is microorganism reproduce asexually. It doesn't fit for microorganism. So we will see the next question, which is, which of the following statement is incorrect or false? Option A, nutrient-rich ecosystem have moderate species abundance. Option B, all cells of the same microbial species show at least 70% DNA hybridization. Option C, the members of the same microbial species have an average nucleotide identity of at least 30% of whole genome level. And option D, the extreme ecosystem have low species richness but can have high species abundance. And option C, Species richness is total number of species or phylotypes present in an area. So among these statements, which is false? Your answers? So I will repeat the question. Which of the following statement is false? Which of the following statement is false? Whether it is nutrient-rich ecosystem of moderate species abundance, or all cells of the same microbial species show at least 70% DNA hybridization, or member of the same microbial species have an average nucleotide identity of at least 30% at whole genome level, or extreme ecosystem have low species richness but can have species abundance. Or species richness is the total number of species or phylotypes present in an area. So which of these is true? Option C. Members of the same microbial species have an average nucleotide identity of at least 30% at whole genome level. Okay, we will see the answer. Yes, the correct option is the members of the same microbial species have an average nucleotide identity of at least 30% at whole genome level, because the members of the same microbial species have an average nucleotide identity of 94% at whole genome level. Now we will see why other options are correct. Nutrient rich ecosystem have moderate species abundance. What does it means? So if we take in an ecosystem with high nutrient, more resource will be available, right? So that supports a large number of individual microorganisms from different species, which resulted in moderate species abundance. And all cells of the same microbial species show at least 70% DNA hybridization, which tells us if two organisms have at least 70% DNA homology, they are considered to be same species. And extreme ecosystem have low species richness because extreme conditions like higher temperature regions like desert region will have low species richness. That is, they will have limited species, but that limited species number will be higher. That is why they have higher species abundance. For example, if we consider the desert region, the uh, if we consider if we consider the desert region, there will be higher thermophilic bacteria will be prevailing in it. So other normal moderate condition or psychrophilic bacteria will be very less. So a single species of thermophilic bacteria will be higher prevalent and their number is higher in number. That's why they have low species richness but have higher species abundance. And as we have seen in the lecture 2, the species richness is nothing but total number of species that is present in the given ecosystem. So the option C is the wrong, which is false. So we will move on to the next question. Which of the following statement is incorrect? Option A, the gills are formed by the organism that are metabolically similar and utilize the same resource. Option B, all organisms living in a specific environment at a specific point of time comprise the population. Option C, the microbial community comprises of multiple guilds. And option D, only phototrophs can synthesize new organic matter from inorganic matter. Option E, allochthonous organic matter is the one which enters the ecosystem from an external source. So you have to identify which statement is incorrect. And your answers are, I will repeat the question once again. Which of these following statement is incorrect? You can view the statements. Gills are formed by organisms that are metabolically similar and utilize the same resource. Option B, all organisms living in a specific environment at a specific time comprise the population. Option C, the microbial community comprise of multiple guilds. And option D, only phototrophs can synthesize new organic matter from the inorganic matter. And option E, allochthons organic matter is that which enters into the ecosystem from the external source. So you can tell which of the following statement is false. So option E is false. Okay. Any other answer? Okay. Any other answer? Which of the following statement is incorrect? Option E. Option E. So we will see the answer now. The answer is D, which is only phototrophs can synthesize new organic matter from inorganic matter. This is not true. Even chemolithotrophs can also synthesize the new organic matter from an inorganic matter. That is why the option D is false. So we will see the other options. As we have know that organism within the gills, they share the metabolic pathway and utilize more or less the same resource. Option A is correct. And option B, all organism living in a specific environment at a specific time comprises the population. Yes. Option C, microbial community means it contains multiple gills. This has been shown in the week 2 lecture. And electron organic matter, which is nothing but the organic matter that is transported into the ecosystem from the external source. So the option D is incorrect because not only phototrophs, even chemolithotrophs can synthesize the new organic matter from the inorganic matter. So we will move on to the next problem, which is the correct order of the metabolic distribution of microorganisms across decreasing gradient of oxygen concentration. I will repeat it once. We have to arrange them in the decreased gradient of oxygen concentration. Option A, obligate followed by facultative followed by microaerophils followed by obligate anaerobes. Option B, obligate aerobes followed by microaerophils followed by obligate anaerobes followed by facultative anaerobes. Option C, obligate anaerobes followed by facultative anaerobes followed by microaerophiles followed by obligate aerobes. Option D, obligate anaerobes followed by facultative anaerobes followed by obligate aerobes followed by microaerophiles. And option D, microaerophiles followed by facultative anaerobes followed by obligate aerobes followed by obligate anaerobes. You just carefully read the question and arrange them in the decreasing gradient of oxygen concentration. I will give you 30 seconds. In option B, it's not specified whether it's a species or not. So here all organism refers to not only species, it comprises of all organism means it can be any species. Even prokaryotes, eukaryotes, algae, plants, animals. So these all comprises of population, right? We have seen different gradients of level. So population, guilds, community, right? So population comprises of all organism. Option A. Okay. Option A. Any other answer? It is a decreasing gradient of oxygen concentration. Option A. Okay. Any other answer? Option A. So before moving on to solution, like anyone tell the definition of obligate aerobes? Obligate aerobes means obligate aerobes. These are microorganism. Obligate aerobes. The term itself telling us that it can survive only in the presence of oxygen. Correct to him. So it needs oxygen. Oxygen is must to survive. So what do you mean by facultative anaerobes? Facultative anaerobes. Facultative anaerobes. The microorganism survives. Yes, it can generally lives in anaerobic condition, that is in the absence of oxygen, but it can survive in oxygen also. So it favors the anaerobic condition, but it can also survive in oxygen also. But it can live in the presence of oxygen. Correct, doing. So microaerophils means microaerophils. The term itself gives the definition microaerophils. That means it needs little amount of oxygen to survive. And obligate anaerobes refers to again, it can only survive in the absence of oxygen. So now we will see the now we understand the different terms and it has been jumbled in different ways. Now we can see the answer. Yes, you are correct. The answer is A. The answer is A, and we will see the definitions in depth. Obligate aerobes are nothing but these microorganisms requires oxygen for growth and will be only found whenever there is an abundant oxygen presence. That is why it is obligate. That is compulsory. Aerobes means aerobic condition. Aerobic condition means oxygen presence. So compulsory oxygen. So facultative anaerobes means these microorganisms can grow with or without oxygen. But mostly they prefer aerobic, sorry, this is anaerobe. So if they prefer aerobic condition, what happens? They can generate more energy through aerobic respiration. So facultative anaerobes are nothing but they can survive in anaerobic or aerobic condition. But if they choose to survive in aerobic condition, then they will generate more energy through aerobic respiration. They will be found in the areas. Sorry, this definition is incorrect. So they will be found in the less oxygen concentration, but can survive in the higher concentration also. So this is the facultative anaerobes. I will repeat, it can grow both condition, but they more prefer anaerobic condition. And if they want to go in aerobic, they will generate more energy. And microaerophils are nothing but they require oxygen, but only in the limited concentration. They will be found in the regions where moderate oxygen levels are present. Obligate anaerobes means these organisms are armed by oxygen. So once oxygen is present, they can't survive. So it is strictly anaerobes. Where Oxygen is completely absent. So the option is the option is option A, and we will see the last MCQ for this session. Which of the following statement about biogeochemical cycle is false? So you have to carefully identify which is the false statement. Option A, all microorganisms can convert inorganic minerals to their bioavailable forms. Nutrient cycling involves oxidation and reduction reaction, and nutrient cycling results in change in chemical speciation, solubility, mobility, or toxicity. Option D, biogeochemical cycles of different elements are interlinked. Option D, carbon is the only major element that can be fixed by higher eukaryotes. So, what is the incorrect statement about the geochemical cycle? I will repeat the question once again. Which of the following statement about biogeochemical cycle is false? Option A. Okay. Any other answer? Option A. Okay. Any other answer? Okay, we will see the answer. Yes, it is option A. All microorganisms can convert inorganic minerals to their bioavailable forms. No, it is incorrect because only some prokaryotes have the capability to convert inorganic minerals to their bioavailable form. And we will see the other statements. Nutrient cycling involves oxidation and reduction reaction. And we have seen in the biogeochemical cycle lecture that microorganism involves oxidation and reduction reaction. So anyone can tell the definition of oxidation? Oxidation means oxidation is a process where what happens? The electrons, will they gain or donate during oxidation? Oxidation refers to the process where, yes, it is correct, twin. It is addition of oxygen and elimination, that is donating the electrons. Whereas reduction means it accepts the electrons. So then in biogeochemical cycle, we observe both oxidation and reduction. That's why we call it carry out the redox reaction. And if you see the nutrient cycle results in, yes, it the impact of the nutrient cycle on chemical form and behavior of elements. And third option, the biogeo cycles of different elements are interlinked because if you see particular cycle, the carbons and nitrogens, they are also closely linked to each other. And carbon is the only major element that can be fixed by higher eukaryotes. Yes, carbon fixation is the one of the major elements or process performed by higher prokaryotics. Carbon, as we have seen, carbon fixation is one of the examples. So carbon is only one of the major elements, whereas the other elements that can also be fixed is nitrogen, phosphorus, sulfur, but it is not commonly observed. So the correct answer is option A, which is incorrect. All microorganisms can convert inorganic to their biomolecule forms. So these are all the MCQs for this session. And I have to thank you all for patiently listening to this lecture. And in this lecture, we have seen the recap of the week 3 environmental biotechnology course. And we have seen the sample MCQs of the week 2 and week 3 of Environmental Biotechnology course. And if you have any questions, you can mail me at ajitvelu284@gmail.com, or otherwise you can post your questions in the discussion forum so that our team will help in solving those queries also. And if you have any queries regarding this week session, you can mail me. And I hope you have all completed your week 3 assignment, which has a deadline of 13th August, I guess. So sorry for the inconvenience. I will share the slide. I hope I am audible and my slides are visible. And what I was saying is, thank you for patiently listening to the lecture. And if you have any doubts, you can post it in the discussion forum, or you can mail me in this email ID. And if you have missed my week 1 and week 2 lecture, you can view in my YouTube channel, which is Ajit Kumar underscore V, or you can access those YouTube videos in the recording sheet that is available in the NPTEL Environmental Biotechnology course website. You can go there and you can identify them. And I hope you have all completed the week 3 assignment, which has a deadline of 13 August, I guess. So try to complete before the deadline. And if you have any doubts, you are free to ask any one of the TA of the Environmental Biotechnology course. And if you have missed out my older videos, you can visit the YouTube channel Ajit Kumar underscore V, or you can access it the NPTEL Environmental Biotechnology course. So, do you have any query regarding this session? Okay, Toyin. So, if you do not have any query, and these sessions are recorded and you can access at any time at your free time. And if you have any doubts, you can again revisit these videos and clarify your doubts. So, thank you all. I think you can leave now, and the session is over. Thank you all.